In this video, I'm going to walk you through the steps you need for the Introduction to Genomics and Galaxy tutorial on the Galaxy Training Network. You're going to do this tutorial for the bioinformatics class as a really nice way to get used to the controls on Galaxy. I like this tutorial a lot because they've done a really good job of setting up practice with clicking the different parts of the window and learning how to set up tools. So I have my Galaxy Genomics genes already run, and I'll walk you through how I got each of these 6 items in my history. So the first thing I did was I needed to get data for where genes are located on chromosome 22. I did that with the Get Data set of tools, scrolling down to the UCSC mains table server. This opens up another window, and a lot of times students feel like maybe Galaxy might be broken, but Galaxy hasn't closed. Anything you do in this window will get imported into your history. We can leave most of the settings on the default settings. The default is to look at the whole genome, but in this tutorial, we're going to look at only the smallest chromosome, chromosome 22. You can click Get Output and then select the whole gene. I can send query to Galaxy. I'm actually going to skip that step and reload Galaxy since I already have the data in there. Once you pull the data in, you'll want to use the pencil icon to give it a meaningful name. I copied what was in name and put it in info. That's something I like to do because it helps me keep a record of how I brought the data in. And called it genes on chromosome 22. You can click the eyeball to see what the data is. A bed file, if you remember from the What the FQ assignment, is a table, and it's just going to list the start and stop of some genetic feature. In this case, we're looking at genes. Every row in the file, and there are 5,351 rows, corresponds with a gene. It shows the start and the stop, and for this tutorial, we really care about the strand. Minus strand genes are reverse genes, plus strand genes are forward genes. So the first thing we want to do with this large file is split it into pieces based on that column. We can do that with the filter tool. So I can find filter by searching for the word, filter data on any column, and I want to look for column 6, that's the strand column, being plus to find the forward genes. For the reverse genes, I want to set column 6 equal to negative, equal negative to check if and keep only the ones where it is a minus or reverse gene. Now that I have the separate lists, I can make comparisons between them. And I'm looking for a function that will help me look for overlaps. The best function for that on Galaxy is called intersect. And I can look in my history and use the run again function to see what the parameters were, or the letter I info to see how I ran this. So if I'm looking for overlapping intervals of forward that intersect with the reverse, I'm going to get a list of overlapping forwards. If I switch the inputs, where I look at reverse that intersect with forward, I'm going to get a list of overlapping reverse. So now I have what I'm looking for, but they're in 2 separate files, and I want to put them together. I can do that with concatenate, the concatenate tool. Concatenate multiple data sets tail to head. Use this tool, not the one that says cat next to it. This is similar to the concatenate function in R or in Excel. In R, we use this function all the time. It's just the letter C. Once we have the final list, I can see how big it is. So it's 2,000 lines. So that means of 5,000 genes, 2,000 of them overlap. And honestly, that seems like a lot. So the tutorial asks us to check it out manually and see if that's true. So when I click on Visualize, I have the option to open up the browser. And in addition to the data that is available on UCSC, there's a black track that's been added here. That's my data from Galaxy. I can go zoom out on the genome and look for a region that's full of both forward and reverse genes. So I'm looking for something that's pretty dense in genes and also is marked on my user supplied track. So I'm going to highlight this here, zoom in. I'm going to keep zooming in until I get to a region where genes are overlapping. So I see a green gene here overlapping with a blue gene here. It's exactly what I'm looking for. And when I zoom in close enough to see the individual exon boxes, these rectangles, I can tell that even though the overall boundaries of the gene are overlapping, the exons really aren't on top of each other. So this might not really be what we're looking for, a piece of DNA that codes for a protein both ways. But it took a while to set up all of those steps. And it's instead of having to manually select all of those parameters again, there's an easier way for us to create a workflow that will be really great to run everything, run steps 2 through 6 as one event, as one application. So if I click on here, I can, sorry, not on the gear icon. If I click up here on history options, I can extract workflow. And I've already done this. And I can look at my workflow for Galaxy genes. I left everything default in this workflow. I kept it all with the same names. The names are just the names of the functions that we're going to run, except for this first one. I clicked on this and clicked edit and changed the label. I like to do this with my workflows to remind myself what kind of file I'm allowed to put into the workflow. So I say input bed file. What will happen to it? It'll get filtered, it'll be intersected, and then last but not least, the two intersections will be concatenated into one data set. When I feel good about my workflow, I can go back to the current history. I don't need to save any of these changes. And I can change it so that I can make a new history where I run the workflow. I also don't want to have to repeat this step with the input data set. I want to be able to copy that to my new history to start it. So I can do that with copy data sets. And I can select just this new data set and then the destination history. You can see the first thing that comes up is Galaxy Genomics exons because I've already created the history. And now, since I've already copied it over, I will just switch to that history. And you can see that the first step is the same as the previous first step, 5,351 regions. In order to go from genes to exons, there's a couple different ways you can do it. You could do a new UCSC search, but what I chose to do was to run this function, gene bed to exon intron code bed expander. I extracted coding exons only, and I did it from the genes file. I then had a new input that I could run a workflow on. So I went back to my workflow, clicked run, and when I clicked run, it gave me the option of everything in the history can be an input. In this case, I set it as exons as the input. And clicked run workflow. I won't run it again because I've already run it here, but it did exactly what the workflow was set to do. 2 filters, 2 intersections, and then concatenate. When you do the analysis on exons, you get a result that's a lot more expected for how many overlapping genes there are on chromosome 22. There's only 12. So this is giving us a more refined list. It's answering a different question, but this is sort of more what we were probably thinking about when we formulated the question in the first place. If you have anything in this tutorial that you need help with, please email me. Otherwise, I think this is a great start to help you get used to Galaxy with selecting your tools, running your tools, and viewing the results of your tools.